Thanks, Selena, and I've got the tough job of trying to keep everyone awake for the next 45 minutes. I'm actually going to keep the presentation part of this fairly short to allow lots of time for questions that if I can't answer, Alina's going to. So here are my disclosures, and I think that it's important to show this because there are some companies that I've consulted for which make drugs and therapies in this space. So today I'm going to take you through, I can't talk about all CLL research, so I decided to take you through really 6 broad types of research and then give you a vignette about how each of these may be relevant to you, and then we can expand upon that in the question and answer session. So I'm going to start off with, I think, what most people think about when they think about CLL research, and that's clinical trials. So does the treatment work? Does it work better than what's already out there, and is it safe? I'm then going to talk briefly about health economics, which is really talking about the public health system. Can and should we afford it? So can we get the treatment to you as patients and have it funded publicly? I'm then going to talk a little bit about patient-reported outcomes research and talking about side effects, and talk about how that's actually quite tricky, particularly for continuous therapy. And then I'm going to talk about what we call qualitative research. And that's more trying to understand people's priorities, values, and barriers to care. And then real-world evidence, which is, it's all very well with the clinical trials, but does it actually work when we give it to the patients that we see day to day in British Columbia? And then finally, we're going to talk about that translational science piece, which is really going from hopefully bench to bedside, so taking discoveries from the biology of CLL and how we can target it more effectively. So I'm just going to give one or two examples of each of these, and obviously that's a very shallow look at quite a deep pool. So we'll start off with clinical trials. Does the treatment work better, and is it safe? So I'm going to take you very quickly through what we call phases of clinical trial, and I'm going to take these one at a time. But when people think about CLL research, they're mostly thinking about these blockbuster phase 3 clinical trials where a comparison has been made of a new treatment strategy, typically with a treatment strategy that's been used today, where we're looking at whether that treatment is equivalent or better, depending on the study design, and a large group of patients looking at safety. And typically, these are randomized, and they're typically quite controlled as to which patients can go into these trials and which patients are excluded from these trials, based on trying to understand how the drug works and how safe it is. In many cases, these trials are run by or sponsored by pharmaceutical companies who are looking to use this evidence to get regulatory approval and reimbursement. But the trial I'm going to tell you about shortly is actually something called an investigator-initiated trial, which tends to answer slightly or ask and answer slightly different questions. So that's what most people think about when they talk about CLL research. But in fact, there's a lot leading up to this. The first sort of trial is called a phase 1 trial, and these are often what we call first in human, where we've come up with a great drug or a great strategy that we've tested often in cell lines and maybe in mice and sometimes in primates, but we now go into humans for the first time, and those tend to be small, although some of them are now becoming quite big because they do all sorts of different arms, but typically 6 to 100 patients, and we're really only asking the question about whether this drug is safe or not, because that's obviously a no-go. If it turns out to have weird side effects that can't be managed, then that drug actually stops at that point and can't be developed further. And sometimes we're also doing what we call dose finding. So we're trying to work out what is a safe dose that, we start to look a little bit at effectiveness, but not really. And those are what we call open label. People know what they're getting and they're not randomized, because we're really just trying to work out whether the drug is safe. The next is called a phase 2 study, Typically, those are also single arm, so just looking at one, everyone gets the same drug, and so it's not randomized. And now we're starting to look a little bit more at effectiveness, but we're typically not comparing it to another drug. I've already told you about phase 3, and the lines between phase 2 and phase 3 are becoming a little bit blurred now, and we're now starting to see larger phase 2 randomized trial. And you say, well, how is that different from a phase 3? And I can't answer that question anymore. But we're getting blurring. And the reason we're getting blurring is that the FDA and the EMA, US and Europe, used to say you have to have 1 or 2 phase 3 randomized controlled trials to get your drug approved. And now they're kind of going, well, maybe a good phase 2 would get you there. So that line is also blurring over time. And then we don't really think about this as a clinical trial, but we then have what we call phase 4, which is where the drug is approved, it's out there, patients are using it in routine practice, and we're gathering the long-term side effects. So it's not like a drug goes out there and then we forget about it and it's just being used. We're feeding back long-term side effects to the FDA, Health Canada, to try and understand what's happening in really big groups of patients. Because even if you're using 1,000, 2,000 patients, you don't necessarily see the really rare things that we accumulate over time. And that can then lead to what we call in the US black box warnings, which is a thing that goes on the box to say, you need to look out for this. This is something that's of interest. So this is the pipeline, and most drugs don't get through to a phase 3 because they're not shown to be safe and effective. So there's a big drop-off over time. So let's look at a phase 3 example. And the example I'm going to use is a study that I really like that was published, actually a couple of months ago, called CLL 17. And this was, as opposed to many big phase 3 trials, which cost a lot of money and are very difficult to run, this was an investigator-initiated study that came out of Germany, but was multinational. And it asked, I think, an important and very relevant question, which is, are fixed duration treatments as effective as continuous treatments? So it wasn't trying to show superiority, better. It was trying to show that it was what we call non-inferior. So is it as good? So what they did is they had 3 arms. They had ibrutinib, continuous therapy. They had venetoclax ibrutinib, again, this time fixed duration. I think it was 12 or 15 months, somewhere in that region. And then they had venetoclax obinutuzumab, again, fixed duration. What I liked about this trial is it had very broad inclusion criteria. So it didn't say you needed to have this, you didn't need to be super fit, you didn't need to be under the age of this or over the age of that. It pretty much included everyone, which is nice because when I see the results, I can be confident that this is what I'm going to see when I actually give it to my patients. And it was well designed and sufficiently large to answer the question. And in fact, in this trial, all 3 treatments were equally effective, and all 3 are funded at BC Cancer. So these are the 3 upfront regimens that we have funded and starting to routinely use at BC Cancer. So I don't know whether you were shown earlier survival curves. I was told by Raymond, do not put these into your presentation. So I deliberately put one on every page. No, I think this is one of 2. But what it's showing here is... No, I'll put in more. So what's shown here is a Kaplan-Meier plot, which is estimating the proportion or percentage of patients who are alive and free from disease. In other words, their CLL is in remission over time. And down the bottom is months. And so if we trace a line up from 24 months up to those curves, each one a different arm of the trial, you can see that they're all sitting on top of one another. So they're equally effective. And what's really nice is those curves are all sitting at 90% or more. So these are all very effective treatments. It gives me a lot of confidence when I recommend any one of these treatments to my patients. So here are the clinical trials that are open at BC Cancer, and I'll come back to one that Alina mentioned earlier in just a second. We like to have a trial that is for patients who are initiating treatment for the first time and also ones that relapse. So our trials at the moment in CLL, the first one is investigator-initiated, which is through the Canadian Clinical Trial Group and I think the Intergroup. It's run by SWOG, the Southwest Oncology Group. And this is a randomized phase 3 study comparing starting venetoclax obinutuzumab early versus starting it when you would standardly start it when you hit those criteria for needing treatment. And these are in high risk but don't have any symptoms patients with CLL. So that's our upfront trial. Our relapse trial is pharma sponsored and it's a phase 1-2. So I told you they're blurring these lines here. Open label study of a BTK degrader in patients whose CLL has come back after 2 or more lines of therapy. And then you heard that there's also a trial using sonrotoclax, which is the, I think it's actually a 3rd generation now, BCL2 inhibitor. And that actually is, I think, opening next week. So I'm sorry it didn't make it onto this slide. It is not quite open, but it will be in a second. Yeah. So the question is, and I get this all the time, somebody comes in, they say, well, I hear that there's this trial. Why isn't it open at BC Cancer and why can't I go on it? And that is an excellent question. The reality is that these trials take months to open and they take a lot of work and effort. So we have to prioritize. We can't have 10 CLL trials open at a time because we also wouldn't then place many patients onto each individual trial and we'd be kind of letting down the people who are actually opening it at our study. But we do prioritize studies because of that. And the way we prioritize it is we want a high quality, relevant question. So our patients are actually going on to and contributing to research that actually counts and is going to change how we treat CLL in the future. If it's a randomized trial, we have to be happy if our patient is put on the control arm. So there have been studies in the past where you look at the control arm and you go, well, that's not standard of care anymore. And I'd feel really unhappy if my patient went on. I'd be very happy if they went onto the experimental arm. That looks great. But how am I going to deal with things if they get put onto the control arm? So we tend to put those, not prioritize those patients, those studies. And then we also have this balance of wanting trials to be open for both newly diagnosed or newly treated patients and those patients at relapse. So we tried to have at least one trial in each of those categories. So we're going to have 2 trials at the time of relapse very shortly, isn't it? Or is sonrotoclax up front? Relapse. Okay, good. So health economics. So then the next type of research is looking at can and should we afford the treatment. So the other question that I'm asked quite a lot is somebody comes in and they have a trial paper in their hand and they say, I want this treatment, it looks fantastic. And I kind of go, well, I'd also maybe like to give you this treatment, but there are steps between the study and actually having the patient available or publicly funded for the patient. And those steps are shown here, and this is a gross simplification of the process. But the first one is we need regulatory approval through Health Canada, and they say, can the drug actually be used for our patients? And they're looking at both efficacy but also safety. So they work a little bit like the FDA, a little bit like the EMA, that they approve the use of drug in Canada. It then goes into a second step, which is about reimbursement recommendation. And this used to be under an organization called CADTH, and they've now changed their name to Canada's Drug Agency. And their question is, should the drug be publicly funded? And they addressed a question or an issue that each province was doing this differently and through their own groups. So if you lived in BC, you maybe could access a drug that couldn't be accessed in Alberta. And I think that that is hugely inequitable. So this organization sat on top of this to make recommendations to the province about what to reimburse or fund. Then we go into price negotiation, and that's typically, designated to one of the provinces to work on everyone's behalf. And then the provinces then decide to make the drug available to patients, and we get a protocol, and we can then use it. I'm going to take my pager off in case that's what's doing it. So the health economics piece really comes into play when we're thinking about making that reimbursement recommendation. So the way that this is done, and it's not in any way perfect, is to look at something called the ISA, which is the incremental cost effectiveness ratio, and I deliberately didn't expand that. So that's the additional cost of the drug versus the current treatment divided by the quality adjusted life years gained. So it's a formula that we argue a lot about in terms of what is the incremental cost, and we also argue about what a quality adjusted life year is and whether that's actually been fairly represented or not. And currently, the number that is acceptable is somewhere between $50,000 and $150,000 per quality of life year gained. But it seems to change over time. So those are the sort of calculations that are being made typically by people within the CDA, but also in terms of the person who's putting it forward to actually decide on whether a drug is going to be recommended for reimbursement or not. But that looks at one slice of the journey. And I don't think that that necessarily sums up what happens over time, because we have to think about sequencing. So if you have CLL, it's likely that you won't just have one line of therapy. Over time, you're going to have multiple lines of therapy. So a nice study was led by Versha Banerjee in Winnipeg, looking at sequencing and cost effectiveness in terms of what treatment should go first in terms of costs and healthcare utilization. And these models use something called a Markov model. And I'm not going to go into too much detail, but it talks about how long people will stay on first line, how long and whether they'll actually need a second line treatment, and how long and what costs are associated with that, etc., etc. And then they build a model based on that. And on the right-hand side, you can see the total 10-year cumulative cost of patients, of a patient population without a TP53 aberration. And there's another plot for if you do have a aberration. And then they've got the different combinations. So on the left-hand side, it would be acalabrutinib, and then the second-line therapy would be venetoclax, et cetera, et cetera. And the bottom line is, and this was a study that was sponsored by the company that makes venetoclax, that if you receive venetoclax obinutuzumab first, then the total cost over time is lower than if you start off with a continuous BTK inhibitor. So this would be used potentially to go to CDA and say, I think you should reimburse venetoclax obinutuzumab because over time, using that first will actually save the country money and we'll be able to afford other drugs down the line in addition. But of course, effectiveness is only part of the story, and what I think is equally important is quality of life and side effects, and we have to think about what sort of research is capturing and measuring these. So typically in these big clinical trials, we record every side effect as you come along, and it's given a grade which talks about how severe it is. And we use a very standardized dictionary, which is the acronym is CTCAE. And then the trial reports out the amount of each grade of toxicity, focusing on what is most severe. So we're particularly interested in very severe toxicity, to make decisions, but also to be able to tell patients what they can expect as they receive the treatment. And this was fine for many toxicities, and it was particularly fine if we were giving short bursts of treatment like FCR, where you'd have quite severe toxicities potentially, and then after the 6 months, your treatment would be done, and then hopefully you'd recover and feel well. But it's not very good at low-grade, grumbling toxicities that we see with continuous therapy. So low-grade diarrhea, if you had that for months or years, is potentially quite debilitating and has a massive impact on your quality of life. So how are we factoring in that, and how are we capturing that? And there's really two ways. One is that one of my colleagues at the Mayo, Geeta Thanarashalingam, didn't pronounce that correctly, came up with a new measure of toxicity, which is called toxicity over time. So that takes into account if you have low-grade toxicity, but you have it for a long period of time, that becomes evident, and we can actually capture that and mark it down in these clinical trials. And the second thing, which seems really obvious, but we don't do it very well, is to actually ask the patients what their quality of life is and to record that. And that's called patient-reported outcomes. So these are captured through standardized questionnaires that are completed by patients at set intervals. We can probably actually do these all the time, even outside of a clinical trial, but we aren't very good at doing that. And part of it is that this takes up quite a lot of time for the patient, and sometimes over time patients get sick of them and don't fill them out. But there are actually a number of different tools, and they can be customized for the study. So there's always a balance between getting all of the relevant information, but also the burden on how long it takes to fill out. And we've come up with innovative ways to do it, like handing iPads to patients as they sit in the waiting room waiting for their appointment. So instead of doing the crossword puzzle or reading a book or getting annoyed, you can be filling out things on the iPad. And so this is an example from the Alpine study, which compared zanubrutinib to ibrutinib at the time of CLL relapse. And they did these questionnaires actually a number of times, but the graph here is looking at baseline versus 12 months. And things aren't quite as big as I thought they would be, but you can see that global health, which is on the top left-hand side, improved with treatment, but there was no significant difference between ibrutinib and zanubrutinib. And then down the bottom there, you can see that diarrhea improved a little bit, but not greatly. And again, there was no difference between the two arms. So we are developing tools and increasingly building these tools into our clinical trials so that we're actually finding out how patients are actually feeling as they go through the treatment, rather than just measuring grades of the nastiest toxicity. So then we get on to what we call qualitative research, which is really the best way of getting at patients' priorities and values. So the aim is to understand what goes into how we make decisions. So what are the priorities, the values, but also barriers to accessing and remaining on care, on treatment. And then this is involving patients, families. We also speak to healthcare workers and also people who pay for the drugs and care. And so an example would be what goes into the decision to choose between time-limited and continuous treatment for your CLL. And we can attack this in a number of ways. Questions and surveys, and I understand that some of them are sent out by CLL Canada. Yep. We also have structured interviews, which can be one-on-one or large groups. And we also have something called discrete choice experiments, where we try and turn this into a very scientific exercise of saying, if treatment A had these characteristics and treatment B had these characteristics, which one would you choose? And then we can ask that in about 50 different ways and come to a conclusion about what are the things that actually feed in to your preference for one sort of treatment versus another. So I think that this is important research, and we're really only starting to do this, I think, in CLL. What about real-world evidence? So I said that I really liked CLL17 because I thought that the way they did the study would translate really well into how we treat patients, but that's not always the case. So BC and BC Cancer is really ideally placed to produce real-world outcomes and evidence. And the reason is that we've got a good-sized population. I think that this is a little bit outdated at 5.6 million. And we have the mandate to provide cancer care, diagnosis, and treatment to all patients across the province. So we can be very good at capturing all of the patients, how they're treated, and what the outcomes are, mostly as a quality assurance exercise to make sure that we're actually delivering the right treatment to our patients. So the provincial CLL database was established in 2007 by Alina, Cindy Toes, Dr. Ramadan, Connors, and Briere. It collates patient characteristics, pathology information, treatment, and outcome. It's Research Ethics Board approved, and the first project they put out involved 100 patients, but I'm now told that it's over 6,000 patients are in the database. So I don't think we're yet capturing everyone across the province, but we're making good progress on doing that. So this is a great resource for working out how our treatments are working. So here's an example from Alina where we looked at how things were going for patients who were treated with ibrutinib. And at the time, it was mainly at the time of relapse because this was how ibrutinib was being used during the study period. And there were two real conclusions from this. Number one, the outcomes looked very similar to the clinical trials, which was very reassuring. So patients were doing well. The other thing, which I think was a little bit of a surprise, but maybe shouldn't have been, was that 35% of patients had stopped using ibrutinib, but for half of those patients, it wasn't because their disease had progressed, it was because of toxicity. It was because they had grumbling side effects that made them miserable enough to actually come off the treatment. So toxicity is really important where you're aiming to give a continuous therapy over many years. So I'm going to end up with a little vignette and discussion about translational studies. And these are trying to understand the biology of CLL and how it can be targeted more effectively. So we've got a pretty big enterprise at BC Cancer biobanking lymphoid cancers, which includes CLL. What I've shown here is probably more relevant to lymphoma, but I'll tell you how we've adapted it for CLL. So we've been building this biobank over the last 30 to 40 years. It was established back in the 80s and 90s by Joe Connors and Randy Gascoigne. And what it involves the database, which I've told you a little bit about, and also the biobank. So we have physicians and consenters and coordinators at BC Cancer Centres, major hospitals, and we reach out to private practices across BC. Clinical data then gets entered into the database of patients and their treatments and outcomes, and also the pathology data. So FISH tests, IGHV results, etc. And during this process, we de-identify people who are in the database so that it can't be traced back to an individual patient and your information is protected. Then we have the biobank side of things, and what I've shown here is more lymphoma relevant, and that's having fresh tumors that are sent in or blood for germline DNA, that's important, and serial samples. And what this allows us to do is to leverage the power of linking samples and things that we do on samples with clinical information and treatment outcomes. And by doing this, we can understand the underlying biology and why sometimes treatment works really well and sometimes it doesn't work as well as we'd like. So back in 2014, we said, well, we actually don't have any CLL actually in our biobank, and we need to change that. So we started a process of bringing in CLL samples and enrolling patients into our biobank. And what's different is that the samples aren't bits of tissue, they're blood. And the blood contains both CLL cells and also normal blood cells. And ideally, we want to have not only all the cells together, but we actually want the different fractions of a pure CLL, that we can be confident that our results come from the tumor or the leukemia, and we want the normal cell fractions as well. So we ask patients who have consented to give vials of blood. We then send a fraction off for what we call flow cytometry, which is how we diagnose CLL. We confirm the diagnosis. We work out what percentage of those cells are CLL cells and what are normal cells. And we also do FISH studies on all of those samples as they come through, so that we know what are the genetic abnormalities in that sample. Then, in our research side, with stem cell assays, and Meryl, who's in the audience over there, we produce 3 types of sample using cell separation and flow cytometry. We have one sample that we freeze down, which is all the cells in the blood. We have one sample that's pure CLL, and we have one sample that's pure normal. And all of these samples are then frozen down and kept in liquid nitrogen, so that we can do studies in the future on these. So far, and I have to say this is, I think, the beginning, rather than a fully accrued bank. We have 204 samples, including 16 from patients where they have been taken at multiple times on the CLL journey. So here are some of the ongoing translational studies, which are all being run by Alina. So I don't know quite why I'm presenting them, but I'm very pleased to. The first one is understanding the genomic alterations in CLL, and we have taken 141 of those 204 samples, and they've been profiled with whole genome and whole transcriptome sequencing. And most recently, this has been done through an effort which is across Canada called the Marathon of Hope Cancer Centre Network, which is supported by the Terry Fox Research Institute. The second study is looking at different mechanisms of resistance to the Bruton tyrosine kinase inhibitors, and this is looking at profiling samples taken before and after progression. And we're using something called single cell sequencing, where we look at individual cells and we can start to build up profiles about why or how these cells are escaping from BTK inhibition. And then we also have a really nice study which is trying to understand how CLL turns into aggressive lymphoma, that thing called Richter syndrome. And we're looking at a very rare but interesting form of this, where the CLL turns into Hodgkin lymphoma. And that's a study that's supported by the Cancer Research Society. So I'm going to take you down one brief story, what I think is, for me, one of the most exciting stories, and that's looking at something called tumor evolution. And this is relevant because it's going to start to tell us how we should use our drugs and in what order we should use our drugs. So lots of us think, I did it up until quite recently, that cancer cells, the malignant cells in the cancer, are all the same. But they're not. They're actually different, what we call clones, which have different mutations. And it's a little bit like, a lot of the thinking that we use when we think about CLL and we think about other malignancies now is we think about, Darwin's finches. So Darwin landed on the Galapagos Islands and he drew, all of these different finches, and they'd all adapted on the different islands, to the different sorts of seeds or insects they could actually get. So one finch, they thought, flew across or somehow got to the Galapagos Islands, and then over time, because of what we call evolutionary pressures of what they could actually eat, they adapted different beaks so they could actually get at these seeds, and so we suddenly see what looks like 16 different sorts of finch, but they all came from one beginning site. And this is now how we think about cancer, that we had to begin with one, what we call transformed cell, and then over time, these cells acquired different mutations, and they exist in the tumor. And why is this important? Well, it means that when we apply a pressure, in this case, treatment, we actually see different populations growing or being eliminated by that treatment. And CLL really is the poster child for this entire area, because you can sample many times because it's a blood draw. It's not going in and taking a piece of tissue. And this research is now revealing how these tumors escape treatment and grow back. So luckily, you've already been shown a little bit of this, so I'm going to go over it again, but some of it will be remembering. BTK is a critical enzyme for CLL cells, and if we block this, the CLL cells die. So BTK inhibitors bind to a pocket in the enzyme that is the pocket that actually does the job. So it's the kinase part of the enzyme. So we have these covalent BTKIs, ibrutinib, acalabrutinib, zanubrutinib, and they bind into that pocket. They block the pocket, and the enzyme can't do its job anymore. One major way that CLL cells escape from that is they mutate a particular, or they make a change in the enzyme that changes this amino acid or part of the protein called C481. And it's kind of like, I think it was explained to you as a parking lot, which was really good. This time the parking lot's got a little bump in it, and the covalent BTKI can no longer bind. And so cells that have that mutation grow out. They become the dominant finch. And we can get around this by using a non-covalent BTKI, such as pirtobrutinib, which can still bind that pocket and block the enzyme. So that sounds great. Why isn't everyone on pirtobrutinib, for example? Well, interestingly, patients treated with pirtobrutinib, actually, what we can see is we look at the sample before and after pirtobrutinib. We see that the cells that had that C481 mutation disappear, but another clone, if the patient relapses, expands out and takes over. And that clone has a different mutation that means they don't respond to pirtobrutinib. And I haven't talked about it, but we can talk about it in the question and answer session. What about those BTK degraders? They're very interesting and they work well on all of these different mutations, but they've started to see people who have resistance to those, and they have mutations that completely destroy the pocket, meaning that not only do they not work, but neither does ibrutinib or pirtobrutinib. So when we think about this, we have to think about a number of things. Should we be giving continuous therapy to all patients, or should we be stopping and starting? Should we be giving drugs in one order or another, thinking about these mutations that come and go? Can we actually use them in order and come back to the same drug over time, potentially? And should we be using them in combinations like the antibiotic, that we use for, or combinations of antibiotics that we use for particularly difficult drugs? So we're learning a lot. We've got a lot still to learn, but I think that this is a very exciting area when we think about how we use our drugs and how we should order them in the future. So a couple of concluding remarks. CLL research, covering a number of domains, has really led to a massive improvement in patient outcomes, not only in terms of progression-free survival, but also in terms of quality of life, really over the last 10 to 15 years, moving from when we had chemotherapy alone to an era where chemotherapy is very infrequently used. I think it's critical that researchers continue to ask questions and to ask and answer questions that are important to patients and their families. We sometimes become a little bit sidetracked in stuff that we find interesting. So we have to be brought back to what actually is relevant. And so we're very keen to have patient advocates who sit when we're building research, grants and things like that, to say, well, that sounds very interesting for you, but how is this relevant to me? And I think that that's really important. And I hope that you can engage in research and how the results are transitioned to patient care. And one way you can do that is by filling out questionnaires that are sent to you that help, for example, medications become available to you during the CDA process. So I'm going to stop there, and I'm very happy to answer any questions. Yeah.
In terms of BC cancer studies that you're doing, how do you feed into or take information from the BC Generations data set as well?
Yeah, that's a very interesting question. So there are a couple of projects that we have intermittently docked with. One was BC Generations, and one was the family study, which looked at things that travel in CLL, multiple CLL, cases within families and things like that. I think that we need to do better at docking with Generations, because I think that that's a really powerful program, and I don't think that we've done as good a job at this point at working with them. Yeah.
Kind of a part A, part B, but how do you get your bio samples into the database? Does your doctor do that for you? Do you volunteer? And how does one get into a trial or get chosen or opt into a trial?
Yeah. So those are two excellent questions. I'll do part B first, just to mix things up. So I think at the time that you are coming up to needing treatment, or you're relapsing and needing further treatment, I think it's a good idea to ask the doctor that you're working with whether there are any clinical trials available. And it may be that the clinical trials that we're offering here might be slightly different than if you're in Kelowna or Victoria, and sometimes patients opt to travel. Typically, there's a lot of visits required in these studies, but I think that the first step is to actually ask your doctor at the time what clinical trials are available and would they be suitable for me, because sometimes we look and we go, actually, what we offer standardly would be preferable in this situation. The other thing is, how do samples make our way into our biobank? And there's a number of different ways, but in most cases we have people who come in and consent patients for research. With CLL it's a little bit different because people are feeding in or have the diagnosis and have been treated in a number of different sites. So sometimes we need to be alerted and reach out. And maybe we can provide information about how to be involved in that. And then we are at the moment being a little bit selective at who's coming into the biobank because of the cost of actually and the effort involved in that processing step. And so I think that Alina is looking for specific types of patients in some situations and sometimes not, and she can answer that. And then typically we want to collect samples at what we think are critical junctures. So when somebody's about to start their first treatment or about to start their second treatment, because part of it is we need a number of CLL cells in order to identify and bank them well. But Alina can comment further about that.
Yeah, great, thank you, and thanks for that question.
Yeah.
So, In general, the biobank has run through the main BC cancer centers, so people who have a hematologist or oncologist through one of the main BC cancer sites. We have a number of members of our research team here. I don't know where they're over there, yes. Fantastic. So they are kind of the engine that helps this run, and essentially we are very fortunate in BC through not just the lymphoma biobank, but actually through other cancer biobanking that happens, that there are research associates at all the sites around the province. So if, and they're kind of in the clinics and they should be aware of patients and trying to approach patients or trying to ask the physicians if this patient would be appropriate for the biobank. For our CLL biobank, as Dr. Scott said, we would love to have everyone in it, and certainly we could gain a lot of information and ask a lot of interesting questions that way, but we've tried to target, right now it's mostly patients who are about to start treatment. So actually anyone who's about to start treatment, whether you're on something or going to switch to something else or about to start your first line treatment, we're interested. And we should provide a way, we'll talk about this at the break and come back to you at the end about a way, if you're not at one of the main BC cancer sites, because we do have mechanisms to get blood from life labs and from different labs. And it sort of depends on the region, and there's some nuances, but we can certainly provide some information for people. Because we know, as Dr. Scott said, CLL is different from a lot of cancers where a lot of people are being treated outside of the main cancer centers at either internal medicine doctors' offices, their GP might be their main doctor, or a hematologist that works outside of the system. Yeah.
I also had...
Just before thanking Dr. Scott, just to mention on the clinical trials question, there are websites that are good search engines for Canadian clinical trials. We featured one recently on our bulletin. So if you go on our CILA Canada website in the bulletins page, and we will eventually have a section on clinical trials, but we're not quite there yet. But there is a good search site. So it's time for it's time for the breakout groups now. So I think we run a bit over time.